And we're back in action. Here, I'll go ahead and delete your battery. All right. So I want you to kind of picture or imagine here in space, we have our 3' to 5', right? It's going in that direction as it assembles it in a 5' to 3' direction. The whole initiation complex got there. Now we have this nice long strand of what we call messenger RNA, ROM. What we actually call that is pre-mRNA or primary transcript. I like primary, but it's easier to say pre-mRNA. Maybe it's not pre-mRNA. So as you see here, enzymes in the eukaryotic nucleus modify pre-mRNA before the genetic messages are dispatched to the cytoplasm for translation. This is really cool stuff. During RNA processing, which is what we're talking about now, both ends of the primary transcript are usually altered as well, and usually some interior parts of the molecule are cut out and then spliced together. So again, the picture you have is this long strand of messenger RNA, right? That's what we just covered. So now we're going to go one level at least, maybe 2 levels into the rabbit hole here. So in eukaryotic cells, it gets very interesting, and I want you to kind of remember what I made reference to once before, and that was at the beginning of the Human Genome Project, we talked about it with the one gene, one hypothesis. I mean, one gene, one protein hypothesis. In that lab study that we did on the metabolic pathway in the pre-lecture that I assigned. So in the fullness of time, as we begin to do this, we expected there to be about 300,000 genes because there's at least 300,000 proteins about in each cell that are performing various tasks. But within the first 5 years, I'd say by 95, 96, that number shot down quickly. We could see that there was really only about 100,000 genes. And already we're going, whoa. As we got to 2000, somewhere between 50 to 100,000. And by 2003, that human genome was done, it was laid out, and it's approximately 32,000 genes. That's it. 32,000 protein coding genes. Clearly, one gene, one protein hypothesis is not the rule. It might be the exception. So how does that happen? What in the, I mean, we know how transcription occurs. Where does all these other protein come from? Good question, right? What did you say?
Modification of the mRNA.
That's going to be the third bullet that we talk about. So first, let's get to that pre-mRNA, and why is that not the final transcript? So another big question is, how much of this gene do we need? How much of it do we need to transcribe? Is RNA always going to transcribe it once the transcription factors bind and we found it? It's like jackpot, I'm going to sit here and do this thing all day. Nope, right? Because we only need so much, let's just say, lactase to break down the milk sugar. None of us, I mean, we drink milk, but not to the degree we did when we were babies, right? The way we did when we were babies. So that gene doesn't really need to be expressed very often, possibly only when you have yogurt, ice cream, milk, or cheese, and to much lesser amounts, right? But if that's not in your diet for the day, or even in the last 3 hours, we need to down-regulate that gene so it's not continually being expressed, and we're saving energy putting it in other places. Does that make sense? How is that controlled and regulated? Well, let's talk about safe passage along with the regulation. But one of those is by the alteration of these the pre-mRNA ends. So we have a 5' cap. That's the shortest name it can be called. It's also called sometimes a G cap, because it's a guanine that's at the very end always. So the 5' receives a modified nucleotide. It's a guanine upside down. And now exonucleases cannot bind to it and snip it. Enzymes are very specific. They go in one direction, which is why exonucleases and endonucleases cannot just do the same thing in the same way. They're specific by design. So a 5' end receives a modified nucleotide. It's a guanine that's upside down. So you have a 5' bound to a 5' instead of 5' to 3', as we're used to. And not only that, it has 3 phosphate groups on it instead of 1. So that's your 5' cap. And then the end, the 3' end of that mRNA transcript gets a poly A tail. It could be anywhere from 50 to 300 adenine long. So poly means many. A's. So these modifications share several functions. They seem to facilitate the export. So the GCAP is kind of like the secret handshake to get through the membrane of the nucleus. Okay, if that's not on there, it don't matter what genes you got on you, Mr. mRNA, you ain't getting through. But I know that it's a control. It's a way to regulate if you're viral RNA trying to make yourself outside or something else, or just not meant to be. So you need that 5' and 3' cap. They protect the mRNA from hydrolytic enzymes. What hydrolytic enzymes could we be talking about? Endonucleases. Any nuclease. Endonuclease, exonuclease, nucleases, period, destroy DNA. So just in your digestive system, you have proteases, you have lipases, you have all kinds of sugar enzymes. These are enzymes for digestion that break down large molecules you consume. The cell has them as well, all right? And so these things are out there constantly degrading. Again, that's kind of a good thing because, again, we don't want to produce that protein forever. And there's a chance that that mRNA could mutate the longer it lives in the cytosol. And you don't want to be producing a protein based on a mutated mRNA. So we want to use it quickly before there's time, degradation, and enzymes that break it down to where the message just becomes nonsense. So these are most enzymes recognized only from the end. So they help, third reason, we'll get back to that. Third reason is they help ribosomes attach to the 5' end in translation, which again we haven't covered. So our picture of a DNA at this point, it's something you would want to be able to reproduce on a drawing for a test, is I'd say draw a segment, a gene, or a coding segment of DNA. This is something what you would want to draw, all right? And normally I do kind of draw it, but I'm just going to go ahead and bluff through it up here. So this would be let's just call it primary messenger RNA. This is your pre-mRNA. As it comes, you have a 5' cap. So this represents the guanine triphosphate. It is a 3', but see it has a 5' cap. So what I want us to realize is that the 5' end is over here. Then you have the 5' UTR, and what does UTR stand for? The untranslated region. And then here's the very first codon. Now, if you didn't know already from the reading or from any of the pre-material, the first codon is always going to be AUG. We can say this because it's pretty true from bacteria to blue sperm whale. It's one of those crazy things. When does school start? August, right? So always starts in August, and that's the way I remember AUG, Aug. That's where the protein or the coding region starts. Let's get a +1. And you come through anyway, here's what would be the coding segment of DNA or mRNA that codes for the protein. Then you have a 3' untranslated region as well. This is really cool. Where do we begin? Now, I'm kind of telling you things that are in relation to a prokaryote. So in bacteria, again, it just stops right here and disbands. But in a eukaryote, the mRNA sees that stop and says, okay. But what we tend to find in genomes of eukaryotes, plants included, is that there will be this segment of very weakly bound nucleotides. When you have a long strand, so here's something I did not cover on day one, which I should have, but that's fine. It's not too big a deal until now, is that, I'll go ahead and draw it. And what I'm drawing between A and T, Taryn, is two dotted lines to represent the hydrogen bonds. Hydrogen bonds are weak, but important. Between G and C, there are 3 hydrogen bonds. So the bond between, let's say, 2 strands of DNA, right, that continued on down, if you have a lot of GC, what does that tell you?
Tightly bound.
Yeah, it's going to be tightly bound, very heat organisms that live, like, who's done, well, it doesn't matter. PCR, for instance, and in biotech, I mean, it takes a certain amount of heat to melt. You calculate the heat needed for PCR in part due to your base pairing. You have to take into account how many GC, because there's 3 hydrogen bonds. That takes a discrete amount of energy to separate. AT, having only 2 hydrogen bonds between them, takes less heat. So if we had 3, AT times 2, that's going to be 6. You have 3 GCs, right? That's going to be 9. So 6 units of energy versus 9 units of energy. And if you give 9 units of energy to something like this, or even this to this, you're not going to get the split between the 2 strands that you need. Anyway, that's just a quick application to the biotech there. But what it says here on the poly A tail is that as that RNA polymerase comes, even though it's affixing it, there's a long strand of AUs and UAs, and those are all double-stranded. It's a weak bond. And so it kind of forms this bubble. And then this little hairpin loop forms. It kind of folds in on itself. Like this is the second or third time in my brain I'm going to write on here. My last board was a smart board, where I could do that. I could write right up. Anyway, a hairpin loop right here. You see the hairpin loop? And then another enzyme comes in and affixes this, catalyzes this poly A tail. All right, so most eukaryotic genes and their RNA transcripts have a long non-coding stretches of nucleotides that lie between the coding regions. These non-coding regions are intervening or interrupting, or what we call introns. The other regions are called exons because they're eventually expressed and they will exit the nucleus. And so what has to happen here is something called RNA splicing. You ever splice wires? And you pull and twist them together, and that's what we're doing. We're taking 2 parts and putting them together. All right, so our knowledge of the pre-mRNA, if you didn't already know, just grew a little bit more. So now look at this drawing, and you see you have still in your pre-mRNA your 5' cap, your poly A tail, your UTRs, but now we have these segments that seem to be nonsense. They were what we used to call junk DNA, right? And this is one of the big answers to the problem, right? How do we have so much DNA and so few genes? Answer, introns, right? So we need to splice these together and snip these out. So the introns are cut out and the exons are spliced together, and that is our final. And this is really cool because what we were doing is we always look at protein structure first, and you can do some reverse genetics. If you know the amino acid sequence, you can work your way back to the DNA sequence. But the mRNA we were finding does not match the proteins, the DNA sequence. And we found where it was, and that is because there are introns or non-coding segments. So splicing is carried out by these structures called spliceosomes, which themselves are made up of several SNRPs. SNRPs are small nuclear ribonucleoproteins that recognize it, very much like transcription factors.
RNA splicing removes introns from pre-mRNA and connects the exons together. Splicing involves a complex called the spliceosome, subunits called SNRP. Each SNRP contains a small nuclear RNA and proteins. Certain sequences are required in intron for it to be removed from the spliceosome. Among the requirements are GU at the 5' end of the intron, 5' splice site, and AG at the 3' end or 3' splice site. A branch site toward the middle of the intron is also required. This sequence contains an adenine that plays a key role in intron removal. The first step is binding of the U1 SNRP 5' splice site, followed by binding of the U2 SNRP to the branch site. Next, the trimer of U4, U6, and U5 SNRPs binds at the intron region, completing the spliceosome assembly.
All right. So a few things here. I'm going to shrink this real quick, just a bit. We'll come right back to it. So this polyadenylation signal, this is a real signal, all right, not just representation. It's AAU, AAA, always there. Comes a few nucleotides, a particular number of nucleotides after the stop codon, before that poly tail is there. As for the intron, and why I say that is because what we saw in the video here is the intron, those first two nucleotides and the last nucleotides of the introns must be there in conjunction with that branching point of the A. You can have these two, the GA, you can have the A, you can have the A's here, but unless you have, and those proteins recognize, we have all three, which is why there's so many SNRPs involved in the process. So right here at the GU and the AG, and this is really cool how we do this. We do single nucleotide mutations, and you just chomp your way down. It's the same way we discovered the promoters. So you have your gene, you just keep knocking out portions of the promoter until the promoter doesn't work anymore, and you're not getting any protein. You know, oh, that sequence was important. And one of those things we've learned was important by removing them is that it doesn't matter what's in here, except that it must have this A in a branch site, and it has the GU and the AG. And the SNRPs know right where to bind, so we know right where to clip. I wouldn't be surprised in many plant diseases, possibly, but for sure in human diseases, there's a few that are due in part to mutated proteins, and the source of that mutated protein is the wrong snipping of an intron. Must be, of course, precise, right? Because you have a reading frame every 3 codons. If you shift one way or the other, you throw off the whole message.
The first step is binding of the U1 SNRP to the 5' splice site, followed by binding of the U2 SNRP to the branch site. Next, the trimer of U4, U6, and U5 SNRPs binds at the intron region, completing the spliceosome assembly. The 5' splice site is cut, and the 5' end of the intron is connected to the adenine in the branch site to form a structure called the lariat.
So just to know that this is an active area of research as well, still, even in molecular biology, is one way, for instance, to have learned it in the book is the U2 binds to that AG first. So you have the two sites first, and the branch point is a third one to be bound. And that loop, first it's bent, there's a bending protein that bends the DNA, and then the branching point is formed. Get that kind of lasso, that lariat that you're seeing here. So that's interesting. When I came across this video, I thought, huh, look at that. Still differences.
At this time, U1 and U4 stairs are relieved, and U6 and U5 stairs share positions. Then, 3' splice site is cut and the exons are connected together. Lariat intron is released along with the remaining parts of the spliceosome. These spliceosome sites later dissociate from lariat, and the lariat will be degraded. The final outcome is that two exons have been covalently connected to each other, and the intervening intron has been removed.
Looks like there's plenty of videos on RNA splicing as well. Are doing good. Where we at? We're almost done. A different layout than it was. All right, so these next few slides just go through the still pictures of what we just saw. So here's our pre-mRNA with introns. We know that there are SNRPs, you do want to know. So I don't have you memorize all the little proteins, but this one's general enough. You do want to know about SNRPs, small nuclear ribonucleoproteins. So it's a protein with an amount of RNA. So this is a fourth, right? A fourth kind of RNA we're not going to talk about. But I do want to say that there's more than 3. But 3 is primary. I mean, for sure you want to know 3. But other proteins must come in. Once they do, so we have the binding, whether the AG first, and then the UG, and then the branch point. Nonetheless, the major spliceosome body comes in once those other proteins have joined, and then we can splice out, cut out that lariat of an intron, which we degraded, and we make this what is becoming now our mature mRNA. I'm not going to play this video, but like I told you, I'm going to put it up there. If you want to go a little deeper with a slightly different kind of animation, this one comes from Oxford. You can click on that one. It's 5 minutes long to explain this processing. So some genes can encode more than one kind of polypeptide. Now we know that, right? How does one gene, I want you to think about this, okay? Given what we've just talked about, how does one gene encode more than one kind of protein? Why can we say, or how can we say the one gene, one protein hypothesis is incorrect? What evidence do we have for that? So we modified, instead of saying one protein, and this might be a terminology thing, we call it polypeptide. That's just another name for a protein, if you didn't know. Just like carbohydrate is another name for sugar, polypeptide is another name for a protein. And so what we've learned is that some proteins are only one polypeptide. Some proteins are 2 polypeptides. Collagen in your hair, in your skin, keratin. That's a tertiary, so it's 3 polypeptides. Hemoglobin, your blood, that's 4 polypeptides. Hormones can be 1 polypeptide. Sometimes a single polypeptide, and what I mean by polypeptide is one strand of amino acids, is enough to do the job, whatever that job is. And sometimes it takes about 2 or 3 or 4 polypeptides to form a full functional proteins. Those are the large ones. Yeah.
What's the distinction between one protein that is made up of multiple polypeptides and a protein complex made up of multiple proteins?
Oh, yeah. So these are, that's a good question. There is a difference. So if it's a protein that's made up of 4 polypeptides, that protein is not a functional protein until all 4 polypeptides have come together. Does that make sense? Whereas a complex of proteins are a complex of functional proteins, like RNA polymerase. It has 4 completed, I would say, or complete polypeptides or protein complexes. Yeah. And that also kind of plays in the fact that, to some degree, protein equals polypeptide, synonymously.
Do we also see proteins that can be one functional protein with this certain set of polypeptides, and then add on one more polypeptide, and it's a slightly different protein?
Yep. So I'm gonna say absolutely, most likely. I always figured so. Generally, you might find there's not a lot talk. There's not a lot, when we look at protein structures, all the ones that we're looking for functional ones, and they seem to be pretty different. There's so much diversity in proteins. You really don't even need them to be that close together in structure. I would say that more would be the difference between 2 versions of the same gene, allelic differences maybe in the protein. So if they're still able to carry the same function out, but in a slightly different way, which is what makes each tree different or each human different from each other. All right, so to wrap up here, let's see. So the answer to the question is in the next 2 bullets right there. How does it help? Well, the variations are called alternative RNA splicing. The number of different proteins an organism can produce is much greater than the number of genes. Say that to yourself. So proteins have this modular architecture, which I don't care about, but I want you to look about right here. So thinking about our gene as we know it now, there's introns, and for a split moment, when we snip it out, you can imagine we have 3 domains after processing. When we translate it, 3 polypeptides. Now, it could very well be that all 3 of these are needed in that specific sequence to make a functional protein, okay? But can you imagine, given what I had just said, if yellow domain 2 wasn't here? We could still have a polypeptide, as you were suggesting in your question, out of just these 2, or a functional protein out of these 2 polypeptides, or out of these 2. Or just put together the same 3 in different ways each time you're making a new. So this is just a very small example, but even from this gene right here, where we have only 3 exons, how many protons can we make? Protons. How many proteins can we make? Right? Does that make sense? Out of 3, how many combinations of 3 can we have? Well, combinations of 3 are just a 1, 2, 3. But then we have combinations of 2, 4, 5, 6, right? Did I miscount already? And then you even have them, what if they function in and of themselves, just as a single polypeptide, 7, 8, 9, right? So you have several different proteins that can be made just from the same. And plants have they don't have an immune system the way we do. This is the way our immune system works, but plants can do things just like this when fighting microorganisms that they don't recognize They combine these things in many ways and hope something works. And whatever works gets recognized. They stop making the other stuff and they assemble only that one. Isn't that crazy? Yeah.
So in RNA processing, are these exons transposable?
Yes. Yes. The application of this is many. Think right now, real quick, of any basic plant function or structure, from cell wall or lignin to respiration or photosynthesis. Think of any enzyme necessary for those fundamental plant-based things. If you know, for one, the protein structure, you can attack it. You have an herbicide, right? If it's specific to that class of weeds or whatever, or you have an insecticide or a pesticide, if you know where in the genome it is, and you're able to locate, you can attack it. And you can render. So the applications are anything from, of course, sprays and chemicals to genetic engineering, whatnot. Once you know the protein structure or the location of the gene, I don't know what you want to do with it. I don't know what you want to produce and with what plant. But your imagination and the system you work with is the limit, right? So a lot of chemicals, as a matter of fact, actually work on the protein synthesis pathway and work by getting down to the, a popular one is cytochrome C in the mitochondria, where ATP is produced for respiration. If you stop a plant, or particularly weed from respirating, you kill it, right? And just ask Dr. Burton, right? How much money a year farmers spend in weeds. That's why there's a whole branch of weed science. So this is fundamental. It helps us, even if you don't remember all these things, to be able to explain at least a little bit to an extension meeting. Getting the nuts and bolts of this when you're selling that product, if you ever worked for Bayer or who else sells that stuff, you know, the more you understand about the chemical and where and how it acts, the more credit you have to either selling, buying, recommending, or not recommending that product. If you're attacking a very fundamental pathway, you say, oh, this is going to kill weeds. Yeah, it's going to kill weeds. And every tomato plant I have in my garden, that attacks what? Where is that chemical used? Yeah, you don't want to attack cytochrome C, right? That's not going to distinguish between a weed and your tomatoes. That's going to kill it all. So you have different classes of these, say, insecticides or herbicides. All right. So there's a little bit of the application. That's translation, where we'll start and pick up with next week. All right. You've been patient.